We have quite a panel here for you. We have 5 speakers and 1 guest panelist. So let's start, and I'll go down the list of speakers in order of presentation. First, we have Dr. Joseph Kotsybar, Regulatory Health Project Manager from the Office of Research and Standards within the Office of Generic Drugs. That presentation will be followed by Calliope Sarago, a team lead and Regulatory Health Project Manager, also within ORS from OGD. Followed would be Dr. Arun Agrawal, pharmacologist from the Division of Bioequivalence 1 within the Office of Generic Drugs. Forgive me, my allergies are kicking up here. Then we will be followed by Dr. Onyeka E. Hezie, Regulatory Business Process Manager from the Division of Regulatory and Business Process Management 1 from OPQ, followed by Dr. Tao Lee Nguyen, Regulatory Business Process Manager from Division of Regulatory and Business Process Management 3. And then joining us for the panel is Dr. Hao-To Lee, Supervisor from the Division of Pharmaceutical Manufacturing Assessment 5 within the Office of Pharmaceutical Manufacturing Assessment within OPQ. And there's going to be a test on all these acronyms later on. Whoever wins gets a mug. So our first speaker, Dr. Kotsybar, please, welcome.
All right, thank you, Brenda, for that introduction. Is everyone still with us? Hopefully, it's been a long day of some really good talks. So as Brenda said, my name is Joseph Kotsybar, and I'm from CDER, Office of Generic Drugs, Office of Research and Standards. And I will be covering my talk today on the overview of the FDA Product-Specific Guidance Program, otherwise commonly known as PSGs. So in this talk today, we're going to cover a few different things. We're going to start by trying to get a good understanding of just the general high level of what the PSG program is. And then we'll talk about its role in supporting abbreviated new drug application approvals. Secondly, we'll talk about the PSG process, the GDUFA-3 commitments, and the adoption of ICH M13A, which we'll talk about on some subsequent slides here shortly. We will identify available online PSG resources and forecasting to help aid in drug development planning. And then lastly, we'll understand how to utilize the PSG public requests, public comments, and GDUFA-3 PSG teleconferences or TCONs in PSG meetings to engage with the FDA. Okay, so we'll start with the basics first. What is a product-specific guidance? So these PSGs, while not legally enforceable, however, they do reflect the FDA's current thinking and expectations on how to develop a generic drug product that is therapeutically equivalent to its specific RLD. These contain product-specific recommendations, obviously that's how it's got its name, as product-specific guidance, to help identify both the methodology for developing generic drugs and generating the evidence recommended to support your ANDA approval. This is a very, these will include both key science and research outputs, including GDUFA-3 research outcomes. And this is a very unique aspect to the generic drug development program. But when you think about PSGs and trying to reach a current bioequivalence approach for a product, it's a one piece of the puzzle. You'll still need to refer always to the regulations 21 CFR 320, which is the section on bioavailability and bioequivalence, as well as the general guidances, which are often referenced in the PSGs themselves. But what was really special about 2024 is there was a very large M13A guidance that required a lot of PSG updates for immediate release solid oral dosage forms. So I'll have a few slides on that coming up shortly to kind of talk about how that work was detailed. So just as like the PSGs are one piece of the puzzle when you think of trying to develop your current bioequivalence approach, the PSGs is just one piece of the puzzle for the ANDA program. So as well with PSGs, there's also pre-ANDA and ANDA meetings that Calliope will be discussing on the presentation right after this. And then there's also controlled correspondences, which we've talked about today through many different talks. But what's really unique about the PSGs is it's really the crossroads of the GDUFA-funded research, as well as the ongoing NDA and ANDA review done at the FDA. So these are basically a development of both those outcomes unilaterally. So some background on PSGs. So the PSG program really began in 2007. The FDA has been publishing PSGs to provide both clear and direct recommendations to ANDA applicants. So as of March 1st, last month, we actually have 2,252 PSGs published. 30% of these are for complex products. So when we say complex here, just a reminder, we're not talking a high-level complex product, but these by definition are complex products based on MAP 5240.10, classifying approved new drug products and drug device combination products. So when you look at the past 10 years of PSG development, it's going to look a little weird. And hopefully if you weren't paying attention to this slide or to my talk yet, this slide probably caught your eye. You're probably thinking, what happened with these numbers? So you can see dating back even before GDUFA 2, the PSG development for both new and revised PSGs have been pretty steady. But here in 2024, you saw about 800 additional PSGs were revised. So again, this is going to be due to the ICH M13A batch that was published in October of 2024, and I will cover more slides kind of on the details of that publication coming up. So when you talk about PSGs, I think the first thing that will need to be understood is the GDUFA commitment to when these product-specific guidances are developed and when applicants should be ready to or should be expecting to see these PSGs published on the website. So for non-complex new chemical entity, new drug applications approved after the start of GDUFA 3, the PSG will be issued for 90% of these NDAs. In practice, that number is if not, I think 100% dating back to the start of GDUFA 2. So the FDA has been very well ready to meet that goal. But what was new with GDUFA 3 is for complex products. So for complex products approved in NDAs, and I'm going to pause there and specify here the difference. These are any NDA approval where non-complex new drug applications have to be an NCE or new chemical entity. That would be a type 1 NDA approval. For complex products in any NDA that was approved after the start of GDUFA 3, FDA will try to get 50% of those products published within 2 years, an additional 25 published within 3 years to hit that 75% criteria. And of course, there's going to be some complex products that were approved prior to the start of GDUFA 3. FDA is still working to get those PSGs published accordingly. So this is a great slide with a lot of information that commonly gets asked how PSGs kind of are developed and how are they prioritized into these batches that get posted. So when we think about the initiating events that kind of lead to a PSG publication, the first and foremost is going to be those recently approved NDAs and supplement NDAs, especially those ones that were approved after the start of GDUFA 3. Those are going to be subjects to the GDUFA goal commitment. However, there's other ways PSGs can be initiated to be developed, such as FDA analysis on products without PSGs, pre-ANDA meetings, public requests, which we'll cover on a subsequent slide, comments submitted under the PSG docket on regulations.gov, controlled correspondence, and citizens petitions. When developing the PSGs, another common question is kind of asked to the FDA, how are these guidances really developed and what data is used? So the data also comes from several different ways. It comes from pharmacokinetic and pharmacodynamic modeling systems, previous BE studies that have been done and submitted in past ANDAs, the NDA review and labeling, pharmacovigilance, and last but not least, the GDUFA-funded research outcomes. When we think about how PSGs are prioritized for publication, the very first, most important one will be those GDUFA commitments I just covered on the past slide. After that, though, the FDA does have and will prioritize PSGs by a variety of ways, one of these being the stakeholder interest in ANDA submission, drug availability and access, public requests from generic drug industry, and public health priorities. So when we think about how the PSGs are actually worked on and then published, the PSG process is extremely cyclical. It typically happens 3 to 4 times a year, and the same process is really carried out those multiple times a year. PSGs are first prioritized for development or revisions. As we just talked about on the last slide, they're grouped together and worked on in parallel, both new and revised PSGs, and then the PSG batch is published. These typically will be quarterly batches or can post as a standalone batch. And we will talk about the standalone batch, most notably with the ICH M13A batch I'll be talking about here in a couple slides. And then after the PSGs are published, they go under a public comment period via the regulations.gov website. Once the comment period's over, these PSGs may be finalized at the FDA's discretion. So if you just noticed on the last slide, I talked about new and revised PSGs. So while most of the slides leading up to this have been really focused on how new PSGs are thought about, initiated, developed, and prioritized into batches, there's also revised PSGs that get posted in parallel. So you may have a question, how do we find or what kind of revisions may happen to a PSG? So there's several ways and several reasonings why a PSG will need to be updated. This could be changes to the RLD labeling, newly identified safety concerns, responses received to SBIA comments, citizens petitions, new bioequivalence approaches, or new knowledge from ANDA assessments and or pre-ANDA meetings and controlled correspondences. When we think about the category of how we group these revisions, you can think about it in kind of 4 big buckets. There's the critical revisions, in vivo and in vitro major revisions, minor revisions, and editorial. Essentially, the difference here is the critical revisions are going to be the kind of most important revision need that's going to affect all incoming ANDAs on a product, all in-house ANDAs, and even past approved ANDAs. That is a very, very rare revision. Most of the time, you'll see either in vivo major, in vitro major, or minor revisions. In vivo and in vitro major revisions are going to require that additional studies or evidence is needed to help support bioequivalence, where minor and editorial revisions are going to be more clarification in nature and more related to just formatting and updating the guidance page itself. Okay, so I know I referenced ICH M13 a few times in this presentation thus far. So now we get to talk about what this ICH M13A batch was. So before that, though, you have to kind of understand what ICH is itself. So ICH stands for the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. No, that's a mouthful. This council brings together the regulatory authorities all over the world and the pharmaceutical industry to discuss scientific and technical aspects for pharmaceutical development. So since 1990, the ICH has gradually evolved and now is developing guidelines for the pharmaceutical sector. Overall, ICH's mission is to achieve greater harmonization worldwide to ensure that safe, effective, and high-quality medicines are developed and registered and maintained in the most efficient manner possible. So for us now, the M13A part is the first ICH generic drug guideline to harmonize BE standards for oral solid dosage forms. So this is a quick snapshot of the front page of the M13A bioequivalence guidance for industry, and I think that's right outside. This guideline ultimately at a high level groups products in 2 ways, in non-high risk products and high risk products. Non-high risk products are typically going to recommend one single bioequivalent study will be sufficient to ensure there is no clinically significant difference in BE under meal conditions. Typically here, you'll see the bioequivalence recommendation being only 1 study, oftentimes the fasting study, due to that being the most sensitive condition to detect formulation impact. In contrast, high-risk products have differences in formulations and or manufacturing processes that may not be detected with a single bioequivalent study, so these will retain both a fasting and fed study recommendation. So why is this important to PSGs? Well, before M13A, there's different regulatory agencies actually had different recommendations for the bioequivalent study design. For example, the FDA required most IR oral products recommended both a fasting and fed study. However, several other regulatory agencies, including the EMA, generally recommended a BE study only fasting conditions. So what the FDA did is, after the final guidance was published in July of 2024, we worked incredibly hard to develop this standalone M13A batch that published just about 3 months after that final guidance in October. To do this, we sought to review all of the necessary PSGs first. So about 1,150 of these PSGs fit within scope of the M13A guidance. Of that initial poll, about 160 of those PSGs did not meet the inclusion criteria. For instance, they may have been a narrow therapeutic index drug, a locally acting drug product, products that already recommended one BE study, or PSGs done or PSGs recommended to be done in patients rather than healthy population. We then took about those 1,000 PSGs and reviewed them for risk. So about 150 of those 1,000 were then excluded due to being high-risk drug products. These included solid dispersions, nanotechnology, retentive formulation, and critical excipients. And there's a great kind of SBIA webinar that was actually done last year in November that gives a lot more background on some of the scientific justification for these updates. So after M13A, so this is the fun slide. So all of those efforts were not for not a good cause. We ended up publishing 814 PSGs in October of 2024. This is a giant standalone batch. So just to reference how many PSGs this is, it's 72% of all of FDA's existing PSGs for oral IR products. Or another way to put it, it's over 1/3 of our total PSGs published in general. That's a very large batch. So the good for industry is that this is going to have significant impact right away. So a quick evaluation was done on the potential cost savings on this. So looking at those ANDAs that were submitted in 2024 with these new recommendations and now the decreased in vivo studies that are recommended, going from 2 studies, fasting and fed, typically down to a fasting study, it's estimated to save almost $50 million per year based on just those ANDAs. And of course, PSGs are continually to be updated and revised. So just because that one M13A batch posted in October does not mean the FDA is done working with some of these PSGs that will need to be posted to update to that M13A guideline. Okay, so now we're going to shift gears a little bit and switch to the online resources and website information. So honestly, the best part or the best place to start looking for FDA PSG information is if you just Google FDA PSG. You're generally going to find the actual homepage for these PSGs that looks like this. At the top of this screen, you'll have a quick link to go to the searchable database to where you can search by RLD, RS number, or API. But if you scroll down, there's a bunch of other really helpful information that will link you to things such as the forecast list, which I'll be talking about in a future slide, the dissolution database, our withdrawn PSGs, and other websites that have resources for the PSG development process. So one question we commonly get is when PSGs get published. So typically new and revised PSGs are generally published quarterly in 4 batches a year. But the FDA can also sometimes post these as standalone guidances or standalone batches, as we saw with the M13A batch that posted last October. Some of the reasons for posting standalone batches include coordinating with the citizens petition response, meeting the GDUFA goal date, efficiency in developing PSGs in the same products class or even the same API, and these level 2 revisions. The FDA also posts in the Federal Registrar for every batch and standalone posting except the level 2 revisions. So you may be asking, okay, so what is this level 2 revision? So when you think about a PSG revision, most of the time in that slide that I talked about earlier, all of those are considered level 1 revisions. These are when the guidance documents set forth in the agency's initial interpretations or statutory or regulatory requirements and describe changes in the FDA's earlier interpretation or policy. Really, the difference here is a level 2 guidance it makes no change to the bioequivalence recommendations. These are going to be typographical errors only, format updates, maybe a change in hyperlink, but these will not update the current bioequivalence recommendation for a specific drug product. So this is also a very helpful resource that I had mentioned on a previous slide, the upcoming PSG for generic drug product development, otherwise known as the forecast list. This may be the best place to go and visit to kind of understand what PSGs are about to be developed. So there are a couple things I want to point out on the forecast list. So sometimes the FDA is unable to directly predict when a batch of PSGs will be posted. So we have to use, to the best of our ability, a within the next 12 months or beyond the next 12 months. We will be looking at the timeline of when we expect these PSGs to be published and update that accordingly. There's also complete transparency in this forecast list, which is a recent addition to the forecast list. So now, every time these forecast list is posted, and we do post this quarterly when those batches typically post, the new PSGs that are posted will come off the forecast list, new workload is added to the forecast list, and any changes that happened from the previous forecast list are highlighted to directly kind of show an updated and change of thinking or timeline. After a PSGs are published, they can go under a public comment process. So the FDA issues a further registrar notice, as I said, for every batch or standalone batch of PSGs. The notice will identify a comment period for the draft recommendations. Comments can be submitted electronically to the docket or by mail. And of course, users can always seek additional assistance with the help desk ticket. FDA does consider these comments on draft PSGs when thinking about if a possible revision is needed. So in contrast, when we think about BE comments being commented on the PSG after it posts, the public requests are kind of essentially the opposite. The public requests are when the PSG does not exist and applicants are looking for that guidance. If you fall into this scenario, you can send a public request for a PSG by submitting to the CDER Direct Next Gen collaboration portal. The FDA reviews these and takes appropriate action. Looking at the past, about 8 years of public request data, I think it's very telling because you see a pretty straightforward decline in these public requests, really since the beginning of GDF 2. I think this is for two reasons. Really just one, just the sheer number of PSGs that have been developed now. There's a lot less need for applicants to submit a public request. But also, I hope, applicants have gotten much better at using this forecast list. So instead of sending a public request, if you go check the forecast list, sometimes that may answer your question about when to expect that upcoming PSG to be developed. Unfortunately, PSGs can and will be withdrawn under certain instances. So these will be withdrawn in recommendations when PSGs are no longer reflect the FDA's current thinking. Withdrawn PSGs could also, and most commonly, be due to when the RLD has also been withdrawn due to safety or efficacy reasons. The FDA does keep this table of all of the past withdrawn PSGs at that initial resource link I shared earlier in the past couple slides. So last but not least, I want to talk about PSG TCONs and PSG meetings, because this is also a very common question we get and something that's new with GDUFA 3. So when ANDA applicants can request a PSG TCON or teleconference at any time during their product development when the FDA publishes a new or revised PSG, if it meets two following criteria. One, the bioequivalence recommendation must be for an in vivo bioequivalence study. And second, the ANDA applicant has already commenced the study. If you fall into that category, you can definitely request a PSG teleconference. Prospective ANDA applicants should submit a request for a pre-submission PSD TCON or a pre-submission meeting electronically through the CDER Direct Next Gen collaboration portal. And applicants can submit a request for a post-submission PSG TCON or post-submission PSG meeting electronically through the Electronic Submissions Gateway. In calendar 2024, PSG TCONs, we received 3 requests for PSG TCONs, 2 pre-submissions and 1 post-submission, and 1 of those PSG TCONs was granted. PSG meetings is basically an add-on meeting after the TCON happens. So this allows a forum to discuss the scientific rationale for an approach or an alternate approach recommended in the PSG. Potential applicants may submit additional questions following the PSG TCON via controlled correspondences, or applicants can also request a pre-ANDA meeting or ANDA scientific meeting as an alternate to the PSG meeting. We will ask, though, that applicants not submit a CC, controlled correspondence, as well as a meeting package or similar for the similar questions. That just creates a little duplicate work for OGD. In calendar 2024, unfortunately, though, we did not receive any PSG meeting requests. So in summary, PSGs reflect the FDA's current thinking and expectations on how to develop a generic drug product that is therapeutically equivalent to the RLD. PSGs are generally published quarterly or as a standalone patch, but you can always find the future forecasting at the link from the FDA website. M13A reduced the need for an additional in vivo BE study for IR oral products compared to what FDA recommended prior to streamline generic drug development globally. And potential applicants are encouraged to submit public requests, request PSG TCONs and PSG meetings, and or leave public comments after the guidances have posted as applicable. Here are some resources, and I'll be sitting at the Q&A panel post all of these talks to address any questions. Yep, thank you. I'll be turning it over now to Calliope.